All right, now that we know a little bit more about how to sample in water and what indicator organisms are and why we care so much about organisms in water, we're going to spend these 3 lectures talking about what's going on in aquatic environments, just the environment as it is by itself, without drinking water or any of the other things that we do as environmental engineers, so that we can understand some of the principles that are really important related to microbes in these habitats. So we're going to talk primarily kind of a high level about habitats, and we're going to take a more time in lecture to go through this in depth. So first things first, what are the major aquatic environments? Well, we have the ocean, obviously, which is a very saline environment that's going to have a huge impact on what microbes live there. We have groundwater, which is going to be important to us as environmental engineers because we think about groundwater wells as a drinking water source. Or if you're into water resources, managing water. We're going to have glaciers and ice caps, and as those melt, we're actually starting to see new microbes coming out of those areas, which is very cool as a new area of study. Snow and ice is going to be a way that we can transport microbes as well, and then lakes and rivers. So lakes and rivers are going to be the big one. When we talk about drinking water treatment, we call lakes and rivers a surface water source. And those are the ones that most of the time we have to spend quite a bit of time disinfecting as a part of our water treatment. So these are the main aquatic environments. We primarily focus on freshwater environments, so things like groundwater, lakes, and rivers when we talk in this class, but that does not mean that marine environments aren't going to have an impact on us. So we'll touch on that briefly when we talk in class. So microorganisms are everywhere, water is everywhere, so microorganisms are going to be everywhere, right? So the total component, if we look at the left side of this screen, the oceans make up the majority of the water, right? That we have is total global water, and then a very small percent of that is fresh water. And then of that fresh water, we move over to that second little block there. A lot of it's groundwater, a lot of it's groundwaters and glaciers and ice caps, and a very small amount is actually our surface water. And then of that surface water or other fresh water, a lot of that is in ice and snow. So there's actually not a ton of water that's available in many ways that is fresh when we think about the total amount of volume of water that's on the planet. So that's just kind of a breakdown of how water is basically balanced on the planet. And so what you can see in the table on the left side of the screen is just the volume, the total volume of that habitat. So 96.5% of the water on Earth is in the oceans, right? And that's a huge impact on microorganisms and what we're going to see. So in some ways, this is actually really good for us as engineers because we know most of the time we're going to deal with fresh water. And we know that that's actually down into kind of the bottom of this table right here. So if we're in a smaller volume of water, it actually helps us start to narrow down the amount of organisms that we're going to have to focus on. So that's actually a huge benefit for us in some ways. So when we define different types of environments or different types of habitats, we have 2 different things that we use. So when we're defining aquatic microbial environments, we can do it by habitat type or microbial lifestyle. So habitat type can be what we call planktonic, and those are organisms that are freely suspended. So exactly what you think of when you think of plankton, right? Floating around in the water. Benthic means that it's associated with sediment. So when we think about the ocean, the benthic zone is at the very bottom of the ocean, right? The benthic zone of a lake is at the very bottom. So these are microorganisms that are in the aquatic environment, but they're specifically associated with sediments, and that's important. And then we can also have what are called biofilms or biomats, and these are organisms that are surface associated. So these are at the top of whatever body of water that it is, but they are associated with each other and they are associated to a specific surface, and that's important. Microbial lifestyles are also going to be important when we think about defining what types of environments we're in. So we're going to talk about primary production and secondary production in aquatic environments in the next pre-lecture. But when we talk about primary lifestyle, we are talking about autotrophic lifestyles. So we're talking about the organisms that can fix carbon from the atmosphere. So they could be photoautotrophs or they could be chemoautotrophs. It doesn't matter which, but we're talking about autotrophy. Then we can also have what's called secondary production, and that is heterotrophic dominant. So we have chemoheterotrophs or photoheterotrophs, but they are responsible for the secondary production of carbon and organics that cycle in a system. So the autotrophs produce the primary sources of carbon that the secondary producers, the heterotrophs, then consume. So both are producing carbon. That's what we mean by production. But who produces what type of carbon is how we do primary versus secondary. And we're going to talk about this more in the next pre-lecture. So don't worry if that's not super clear yet. There are a couple factors that I want to make sure that we talk about so that we're ready to go when we get into class. And one is light. So depending on what type of turbidity we have in our systems, light can penetrate actually quite deep depending on where we are in like what type of environment we're in and what type of water quality factors we have. So we can go as deep as 200, but that's going to depend on the turbidity. And the depth that the light penetrates defines what's called a photic zone. So photo meaning light. So the photic zone is really important because the photic zone, if you think about it, is where you're going to find your photoautotrophs and your photoheterotrophs. So that's really critical because we're not going to have photoautotrophs and photoheterotrophs deep in water, right? So we have what's called the photic zone, which tends to be on the top, and then the aphotic zone, which is below it. And the aphotic zone, remember, A in front of anything means not. So there's no light there, or there's not enough penetration for those organisms to live. So what that means is that in water, we're going to stratify. We're going to have a photic zone where the phototrophs are going to want to live. And then we're going to have the aphotic zone where we're going to have heterotrophs living. And that's going to really matter because the type of organisms that live in those habitats is very different. So that's why light penetration is going to matter to us. So we just mentioned that. I always give you the answer before I come up with the answer. Last thing that I want to talk about is depth. So we have what we call benthic zones, and those are sediment-associated zones, and then we have what are called pelagic zones. It always takes me a second to say this word correctly, pelagic zones. And it's a broad term used to describe the water column or the planktonic habitats. So remember, benthic is associated with sediments. So benthic zones are when we have things like the continental slope and the rise and trenches and things like that. And then the pelagic zone is like the free water, right? It's the water column itself. And so plankton can be anywhere in there, but then we have slightly different zones as we go into deeper and deeper layers when it goes from photic to aphotic. So what you'll see on the left side of this is you'll see just an example using the ocean. And you have different words for different areas, but generally the top layer is called the sunlight zone, and then underneath is the twilight zone, and then it moves down into areas that are completely aphotic, right? They don't have any light penetrating. So that's important. So we're going to talk about that a little bit more when we talk about freshwater versus marine and why this matters. And then the last thing that I want to just draw your attention to on the right side is that that's going to start to matter for what types of layers we have when we have mats or biofilms in a system. So we're going to have air, we'll have some kind of a water lipid layer, we'll have some protein polysaccharide layer, we'll have different layers that form. And that's kind of the same idea as this benthic versus pelagic zone is we're going to have layers that are forming basically all the time in water. So that's just enough to get you started on microbial habitats and start to think about this. We'll talk a lot more about this in depth in class. Your next pre-lecture is going to focus on primary production and secondary production.